A karyotype is an organized photograph of chromosomes. And on a karyotype, the homologous pairs are ordered by size and by the position of the centromere. Karyotypes are used to determine an individual's chromosome pattern. And when a karyotype is analyzed, length, position of centromeres, banding patterns, and other physical characteristics of the chromosomes are looked at. For a karyotype, first, a picture of all of the chromosomes are taken, usually of a cell that is in mitosis, and then it is then organized into an organized pattern, largest chromosomes to the smallest chromosomes. Chromosome number is referred to as 2N, and remember, human chromosome number is 46. So 2N is 46. What that tells us is that we have 23 pairs of chromosomes. Of those 46 chromosomes, some of those are considered to be autosomes. Autosomes are chromosomes containing genes for non-gender related traits, and they're numbered in relation to size. Chromosome 1 is the largest, and that has approximately 2,800 genes, and that's on each of chromosome 1. Chromosome pair 22 is the smallest, and each of those chromosomes have approximately 750 genes. We have sex chromosomes, and those are our two chromosomes that determine gender. XX indicates female, and XY indicates male. In addition to carrying information for all of the gender-related traits, sex chromosomes also contain genes for some non-gender related traits. And the Y chromosome is much smaller than the X chromosome, and it's actually missing some of the information that's found on the X. In terms of patterns of inheritance, Mendelian genetics follows Mendel's rules of inheritance on the autosomes. So on those 44 non-gender related chromosomes, inheritance follows plain old Mendelian genetic rules. Sex linkage is just looking at the X and the Y chromosome. And sex linkage follows Mendelian rules on those two chromosomes, but because we're just looking at those two chromosomes, we're going to see different inheritance patterns. The commonly studied sex-linked traits are colorblindness, hemophilia, which is when your blood lacks clotting proteins, baldness, and muscular dystrophy. And that's a disorder where the muscles basically wither and break down. Here we have the pseudoisochromatic plate Ishihara compatible PIP test. That is the official scientific information or name of a typical colorblind test. Colorblind tests are used to determine if an individual is colorblind. And when an individual is looking at a colorblind test, they should see different numbers or images within the image that they're shown. There's different color patterns because there's different kinds of color blindness. And so the different color patterns that are used are being used to determine if there's a specific type of color blindness an individual may be afflicted with. There's also colorblind tests for children, and especially those that maybe don't know their numbers, but they're able to identify pictures. And as I said before, there are different kinds of color blindness. And so The different kinds of color blindness allow an individual to see colors a little bit differently than normal. Most individuals who are colorblind actually can see color to some degree. It is very rare for an individual to be totally colorblind and see everything in black and white. As you can see, that is about 0.00003% of the population, which is a very, very tiny number. So, for those of us who aren't colorblind, it is kind of hard to understand what colorblind people may see. So these are some examples that might help us understand what they see. The different pictures are showing the different kinds of color blindness and how they would appear to those individuals. Now, one thing to remember about color blindness is that individuals who are colorblind are born that way. They don't know what normal visioned individuals are able to see. My cousin didn't know he was colorblind until he actually took a test for color blindness when he was in 8th grade, because he learned that what he was seeing were certain colors, but he didn't realize he was seeing them differently than individuals that were not colorblind. Now, sex-linked genes and sex-linked traits, the genes are only found on the X chromosome. There's no gene on the Y chromosome for these traits. So, like I said before, that the Y chromosome is missing some information, this would be the information that's missing. These genes are recessive. These traits are more common in males. And that's because males only need one recessive gene to end up having the recessive trait. And that's because this information's not on the Y chromosome. So whatever is on their X chromosome is what will be expressed. So if they've got a recessive on that X chromosome, they are going to show the recessive trait. Females, on the other hand, need two recessive genes to have the trait. If females have a recessive on one of their X chromosomes, they still have a possibility of ending up with a dominant on the other X chromosome and not showing the recessive trait. With sex-linked genes, we need to write the genotypes differently. We need to include the sex chromosomes, which shows us that we're dealing with a sex-linked trait. Then we also have to include the dominant or recessive alleles, like we would with normal Mendelian inheritance. And we need to remember that there's no gene for these on the Y chromosome. So here is an example of how we would write genotypes for color blindness. And for each of these genotypes, you can see what gender the individual would be and what trait they would end up having. So here is an example problem. A colorblind man marries a female carrier. So we need to determine what would the genotype of the man be, what would the genotype of the woman be, and what kind of possible offspring would they have. So here's a Punnett square that shows that. Our colorblind male is at the top of this Punnett square, X, little b, and Y. Remember, we don't write anything on the Y chromosome. The female's a carrier. And remember, carrier is another word for hybrid or heterozygous. So her genotype would be X big B, X little b. And then we would fill in the Punnett square just like we're used to for all of our other sorts of genetic problems. We have to make sure that we keep the X and its B with it. So that's how we would fill in the Punnett square. What that tells us is that these parents could have a normal visioned female, a colorblind female, a normal visioned male, and a colorblind male. So it's possible for them to have any type of children with color blindness or not color blindness. Here's another problem. This time we have a colorblind woman who marries a man with color vision. Again, we need to determine the genotype of the man, the genotype of the woman, and their possible offspring. So when we fill out a Punnett square for this couple, we have our colorblind woman over here. For her to be colorblind, she needs a recessive allele on each of her X chromosomes, and the male has color vision. And so he is X big BY, because remember, there's no gene for this on the Y chromosome. And so then we can see what children they would possibly produce. If they have children, every child that is a female will have color vision, and every child that is a male will be colorblind. What's interesting about this is that the children will have opposite traits that their parents had. So if we look at the two Punnett squares that we just completed, and we look to see who girls inherit these traits from, girls inherit these traits from both parents. Boys inherit these traits only from their mothers. So right here we have a female who is colorblind. For her to be colorblind, she had to get an X little b from her mom and an X little b from her dad. Whereas here, when we have the colorblind male, he's male because he got a Y chromosome from his father. But he's colorblind because he's got the X little b from his mother. So we can also write genotypes on sex-linked pedigrees. And this is similar to what we were doing on regular Mendelian inheritance pedigrees, but there is a little bit of a difference. So we have to remember to use X's and Y's as well as all of the individual alleles for the genotypes, just like we were doing on those Punnett squares. We write all the recessive genotypes first, and we do that for both males and females. And we do the recessives first because there's only one possible recessive genotype for a male, and there's only one recessive genotype for a female. Once we have all the recessives done, sex-linked pedigrees have one additional step that we squeeze in here. After the recessives, we do all of the male genotypes that are remaining. All of the male genotypes that are remaining are going to be males who are dominant. There's only one possible genotype for a male who is dominant for a sex-linked trait, and that has to be X big letter Y. So we want to do all of those genotypes next. So then what remains are dominant female genotypes. And so we finish off the pedigree by filling in those genotypes. And again, if we can't determine if a female is homozygous dominant or heterozygous, we write both genotypes. So here's an example to start. We're going to use the letter R. I just randomly picked a letter. And the ones that are shaded are recessive. Okay. So we need to write the genotypes with the X's and the Y's and the R's, and we start with the recessive individuals first. So the males, to be recessive, they'd be X little r Y. And then the recessive female is X little r, X little r. The next step would be to do any remaining males, which we don't have any. We've already done all the males because they were all recessive. And so then we would do the dominant females. And this female, we know she's dominant, but she has had recessive children. So that means she has to be a carrier. She has to be X big R since she's dominant, but she's carrying an X little r, which is why the children ended up with whatever this recessive trait is. Here's another example. We're still going to use R's. We're still going to say the red ones are recessive. So we start by doing all the recessive genotypes first. Our female would have to be X little r, X little r. And our males would have to be X little ry. The next step is to do the males that remain. So the 3 males that remain are dominant, and the only dominant genotype for males would be X big R, Y. So now we have the 4 females that are dominant remaining. This one here, she has to be a carrier. She has to be X big R, X little r, because she had a recessive mother. So all that her mom can give her is one of those X little r's. Her dad gave her the X big R, which made her dominant, but the X little r came from mom. The other one down here that has to be heterozygous or a carrier is this one, X big R, X little r. We know this because she had a recessive dad. All that her dad can give her is this X little r. Because if dad were to give her the Y chromosome, then she would have been a male. So now finally, those 2 that are left are 2 that we can't tell exactly which dominant genotype they should be. And so we would put both of the dominant genotypes for those 2 symbols.